Alright, and so now it's time to come back to finish up the enzyme chapter, or at least some more stuff about enzyme. Remember, I have a video already on the channel about the Michaelis Menten equation. I also have one about the different types of inhibition. And so what we're going to talk about is enzyme regulation, as well as a little bit more information about what Michaelis Menten, the equation, tells us. And so we know that V is equal to Vmax times S over Km plus S. And so what happens is let's say there's a situation in our body where Km, right, Km is equal to half Vmax, as we saw on the Michaelis Menten equation. And so what we can do is Km is equal to S. Therefore, I don't know why I drew that V like that. Velocity is equal to Vmax S over S plus S. So this is 1/2 V max is the velocity, right? So when Km is equal to S, V, the velocity is about 1/2 of the Vmax, right? And if you were to draw out the Michaelis Menten equation, that is what we see there is that Km. And so in cells, so when you study cells and you study out their substrates and you study out the concentration of their substrates, it maintains a substrate to Km ratio of about 1, right? And so what we can do is we can measure the Km value of a variety of these different enzymes, and then that suggests to us that the cellular substrate concentration then is near that Km value. And so because this is the case, right, because the cells do this, right, where they keep the ratio of the substrate concentration divided by Km is about 1, because of that it's tunable. We can tune up these enzymes. We can drop the substrate concentration, we can increase the substrate concentration for a variety of different things that we might need. So that's the first one, right? So that's the first one, is when Km is equal to the concentration of the substrate, we get half Vmax, and so then we can analyze Kms and enzymes in that direction. But what happens if Km is much greater than the substrate concentration? And so what happens is V is equal to Vmax. I'm going to draw it like this. Km, right? So remember, we have V is equal to Vmax times S times Km plus S. Well, if Vmax is much greater than S, that's pretty much going to be Km. And so then I'm going to draw this slightly differently to where we have this, where it's Vmax over Km is equal to S, or multiplied by the concentration of the substrate, right? This is a first order rate equation. And what we can do, and so when we go back to the Michaelis Menten and we see what Vmax is, what we can get Vmax is the catalytic rate constant times the total amount of enzyme divided by the Km from up above times S. And what we can do is let's split these up even one more time so that we get velocity is equal to K cat divided by Km times E total times S. Right? And this right here is known as the specificity constant. The specificity constant, and it's a way for us to compare different enzymes, right? It's a way for us to tell how well an enzyme works, and we can compare multiple different enzymes together. And so what happens is, if a specificity constant is around 10 to the 8th or 10 to the 9th, we call that enzyme catalytically perfect. And what we mean by that is that the diffusion rate is about 10 to the 8th or 10 to the 9th inverse seconds. And so what happens is, as soon as this enzyme sees its substrate, it acts, and it turns it into product. And so there's two examples of these. One is catalase. So catalase is an enzyme that turns hydrogen peroxide. It breaks down hydrogen peroxide in our body to water and oxygen, because we know hydrogen peroxide is very toxic in our body. It can oxidize a lot of different things. The other one is carbonic anhydrase. And this takes CO2 and turns it into bicarbonate, and is very, very important for our buffering system. Right? So that's if the Km is much greater than the concentration, is we can calculate that specificity constant. And so then what happens if the Km is much less than the specificity constant? Well, if we look at our Lineweaver-Burke plot, we're going to be out here where the velocity is equal to the Vmax, right? So we're going to be at saturation levels, and so when we see the velocity, that is going to be Vmax. So let's talk a little bit more specifically, I'll just do it on the other side here, of turnover number. Right, so we're going to look at kcat for this. And so we want to know is how much a substrate is converted to product. And so it's number of substrate to product per second under saturating conditions, right? So we're saturating the reaction with enzyme, and we want to know how much product or how much substrate is converted to product. So Vmax, we said, was equal to kcat times total enzyme. And so let's solve for kcat. Kcat is equal to Vmax over enzyme total. Now, carbonic anhydrase. So we said carbonic anhydrase is a perfect enzyme. Its E total is 1 times 10 to the negative 6th molar. Its Vmax, if we calculate it, is 0.6 mole per second. And so we know that the kcat is 0.6 divided by 1 times 10 to the negative 6th. It's going to be 6 times 10 to the 5th inverse seconds. And so per second per molecule enzyme, carbonic anhydrase can convert 600,000 molecules of CO2 into bicarbonate. Now, what we can do is we can do 1 divided by kcat, and that'll give us the number of seconds per molecule, right? So this is molecules per inverse seconds, or per second, but if we take the inverse, we could do how many seconds per molecule when you do 1 divided by this number. Let's do it in a different color. When you do a 1 divided by that number, you get 1.7 times 10 to the negative 6 seconds, or 1.7 microseconds. Right, so it takes carbonic anhydrase 1.7 microseconds to turn a molecule of substrate into product. So, we have a video that's already out there about chymotrypsin. Chymotrypsin is a protease. It degrades proteins in our body naturally, but we don't want those proteases to be active all the time in our body, because not only will it inhibit and break down our dietary proteins, but it'll also inhibit and break down our proteins as well. And so the gallbladder and pancreas, the pancreas releases these proteases, these zymogens, but they're also important for the inhibition. And so what happens is there's a thing called antitrypsin. Right? And so what antitrypsin's role is to inhibit trypsin. It's a pancreatic trypsin inhibitor. The KD, the dissociation constant of this inhibitor, is 0.1 picomolar. So it pretty much binds and does not release, which makes sense because we don't want these proteins constantly active. And so what happens is This is the anti. And here's just the trypsin. And there's an active site carboxyl group that's in there, right? Part of that catalytic triad we talked about. Well, antitrypsin, I'm drawing them as solid lines, but these are H bonds. Has a lot of hydrogen bonding with the trypsin. And there is 2, 3, 4 a lysine residue that kind of fishes into the active site. And what happens is that forms an ionic bridge to even further stabilize this interaction. This active site having this carboxyl group is known as an oxyanion hole. Right, and within this hole there's a negatively charged oxygen that this lysine side chain can fit right in there, form an ionic bond, and that's why the Kd of antitrypsin is so low. So there are proteins, we haven't talked about them yet, but there are proteins that phosphorylate. These are known as kinases. Right? Kinases put a phosphate onto things. Kinases are very important signaling molecules or signaling enzymes in our body. They amplify signals that they get, and they can either turn things on or turn things off depending on what they are and which one we're looking at. So let's look at a kinase known as protein kinase A, or pKa. So what happens is in the bloodstream, you have one molecule of hormone, or a neurotransmitter, or some other signaling molecule. There's two ways that this can happen. Here's the membrane. There is a protein in the membrane known as adenylate cyclase. Right, this hormone can bind to there, or it can just go straight through the membrane, and so things like estrogen can pass through the membrane to estrogen receptors within the cell. And what adenylate cyclase does, though, if that hormone signal binds to it, it turns ATP into a molecule called cyclic AMP. I would recommend looking up the structures of ATP and cyclic AMP. I don't have enough room to draw them on here, just so that you can be familiar with it. And what happens is the signal is increased, right? So we're only talking about one molecule of hormone. Adenylate cyclase will make many molecules of cyclic AMP. What cyclic AMP then will do is it will come and it will meet up this little puzzle piece. And this puzzle piece has 4 binding spots to where cyclic AMP can go. And all 4 spots have to be filled with cyclic AMP for it to release our pKa. Right? So protein kinase A is normally inactive in our cells. And what happens is this protein kinase A's can phosphorylate. They put a phosphate group on serine, threonine, or tyrosines, right? Anything with that hydroxyl group. All 4 of them have to be bound, and then it can release the pKa, right? And so once the pKa is released, it can go off and phosphorylate. And so what does it do, right? What do these kinases do? And so here, let's look at this. So here's a serine residue, and some kinases can do serine, some can do threonine, some can do both, some can do tyrosine only. It depends on the enzyme and its active site. Whoops. I'm just going to draw a phosphate like this. So whenever I draw a phosphate like this, what it means is that it's this. Right? It's that phosphate group. I just don't want to draw everything just for the sake of space. And so going from the left to the right, we now phosphorylate it. That's a kinase. Kinases use ATP, turn them into ADP. So this would be pKa, right? This is what pKa would do. pKa would phosphorylate proteins. This activates some proteins. This inhibits, stops the function of some proteins. This blocks two proteins from interacting with each other. This allows two proteins to interact with each other. There's a lot of things that phosphorylation does in the cell. And then what we can do, with a little bit of water, we can take that phosphate group back off. This is known as a phosphorylase. Right, so phosphorylases can remove phosphates groups. Not all serines are phosphorylated, not all threonines and not all tyrosines. They have to be known and they have to be found in what's known as a consensus motif. And this consensus motif for pKa is arginine, some X, which is usually a small amino acid like a glycine or an alanine, and then you can have a serine or a threonine, and then some Z, which is always a hydrophobic. Z has to be always a hydrophobic amino acid, right? And so when pKa senses or sees, quote unquote, this sequence, it'll phosphorylate it. So we said in the previous slide, pKa is currently being inhibited by this inhibitor. So the question is, what type of sequence do we think is right here, where it's bound to pKa? If you think saying, oh, well, it should probably look like a serine or a threonine, you're right. And here's what it's known as a pseudosubstrate. Right? It's a pseudosubstrate inhibition. And what we have is an arginine, a glycine, right? So a small amino acid, and then an alanine, and then an isoleucine, right? So here is the difference. In the real stuff, in the real substrate, you have something you can phosphorylate. In the pseudosubstrate inhibitor, we have something that mimics it, but it cannot be substrated, or cannot be phosphorylated. And because of that, it inhibits. It's why that the inhibitor is bound. We're not going to talk about it, but if you want to, there are some videos on glycolysis already posted that look at feedback inhibition. Look at how some enzymes can actually be inhibited, by downstream products of the reactions. And it's a very common way. We'll also see, we'll also see that the first step of any chemical reaction is probably the most regulated. Because if we can't start it, then we don't have to worry about all the energy it takes to do it.